this segment we're going to talk about digestive physiology. So we're really talking about digestion and absorption. Digestion is the chemical breakdown of macromolecules. Absorption is how do we absorb them into our body once they've been broken down. The digestive system first breaks down those materials that we ingest, and then we'll disassemble them into smaller fragments. Those released molecules will create ATP once they're in the body, or we can take those smaller fragments and make fats, proteins, and carbs in our body. Now, what we need to know first is the breakdown of each of these macromolecules. So let's just talk briefly about that. Carbohydrates, and we know carbohydrates come in the form of whole wheat, veggies, fruits. Okay, so when we ingest these materials, we're ingesting actually a polysaccharide. So that is a large carbohydrate. And those polysaccharides then will get broken down into smaller units, which are called disaccharides and trisaccharides. And then those di and trisaccharides will need to be broken down into very simple sugars. And the simple sugars we call monosaccharides. And it's these monosaccharides that we are able to absorb into the body then, and then they can be used to create ATP. The most common monosaccharide is glucose. Another common monosaccharide is fructose. Okay, so those are what we're looking at when we're talking about carbohydrates. Lipids, on the other hand, lipids are going to enter our body as larger triglycerides. Okay, and we can get these lipids from foods like animal products, such as dairy. Okay, some fats, things like that. The triglycerides are going to get broken down into smaller units, such as fatty acids and glycerol or monoglycerides. And then we'll be able to absorb those into the body as well. Okay, and then finally we have proteins. Okay, and proteins again we can get from meats, eggs, foods like that. And proteins start out as large chains of amino acids, and proteins will get first broken down into polypeptides. So they're little shorter chains of amino acids, and then into dipeptides, so shorter yet, and finally into the individual amino acids. And it's these amino acids then that will get absorbed into the body. Okay, now that we know all of that, we're going to start out with carbohydrate, digestion, and absorption. So the first thing that happens is that we ingest the carbohydrate. So we bring it into the mouth. That's called ingestion. Once in the mouth, these salivary glands will start secreting a substance called salivary, because it's coming from the salivary gland, so salivary amylase. Salivary amylase. Now, salivary amylase is going to start to break those polysaccharides into di and trisaccharides. Okay, so we're going to break them down a little bit smaller. And then we swallow. and as we swallow, those polysaccharides, because there's some that haven't been broken down yet, and the di and trisaccharides enter into the esophagus. Now in the esophagus, this is just a tube here that there's no secretions that occur in there and no absorption occurs there, but it is a tube where the continuation of the digestion of these polysaccharides can continue. And so that's what happens. These polysaccharides continue to get digested as you're swallowing, and then they get down into the stomach area. Now, in the mouth and in the esophagus, the pH was at about 4.5, and that's a good pH for this salivary amylase to work. But once we reach the stomach, there's a lot of other acids in there, and the pH will then drop to 1.5 to 2.0. And this happens to be too acidic for the salivary amylase to continue working, so we kind of get a halt of the carbohydrate digestion in the stomach. So it stops. But then the chyme, after the stomach churns it up and mixes it with the enzymes in the stomach, the chyme will enter into this longer tube, which is called the small intestine. And the first part of the small intestine, of course, is the duodenum. And in the duodenum, there will be some secretions from other accessory organs. One of those accessory organs is called the pancreas. And so this pancreas, which is right here, okay, pancreas is going to deposit some of its secretions into the small intestine, into the duodenum. And one of the substances that the pancreas secretes into the small intestine is called pancreatic amylase. Okay, pancreatic amylase. And what pancreatic amylase does is it breaks down polysaccharides into di and trisaccharides. Okay, so we're going to end up with these di and trisaccharides. All right, because pancreatic amylase can work at a lower pH. Okay, now if you figured this out, amylase means that is an enzyme that breaks down carbohydrates. All right, now that we've broken down those polysaccharides into di and trisaccharides, we are looking to see if the di and trisaccharides can get absorbed into through the intestine and into the body, and actually it can't. It has to be broken down even further. So there are some, we know in the villi, in the small intestine, so there's these plicae, which are folds, and on top of the plicae there are these villi, and then on top of the villi there are these even smaller finger-like projections, okay, that are called microvilli. And the microvilli form kind of a brush border, we call it. So it's a brush border in the small intestine. And the brush border, the cells lining the brush border, are going to secrete an enzyme, or secrete enzymes that are called brush border enzymes. And it's these brush border enzymes that will break down the di and trisaccharides into the monosaccharides. Okay, and it's these monosaccharides that can get absorbed into the body. Okay, so now we have monosaccharides. We've been able to break those polysaccharides all the way down into those small monosaccharides. The monosaccharides are going to get absorbed in the jejunum, and the jejunum is the second part of the small intestine. So here's the duodenum. It's very short. It's only about a foot long. And then we have this long jejunum. And the jejunum is there are these again villi, and inside the villi there are some blood vessels. Okay, and these blood vessels lead into larger blood vessels, and these larger blood vessels are going to head to the liver. Okay, so monosaccharides get absorbed into the blood, and all blood leaving the intestinal area will go to the liver. And here in the liver, the liver will do some metabolic regulation and figure out what to do with the monosaccharides. Okay, the next thing we have are lipids. And lipids are going to enter into the duodenum, that first part of the small intestine, as triglycerides. Okay, so what does that tell us? Well, we know we ingest the lipids, so nothing really is going to happen in the mouth. There are some salivary lipase that will start to break down those triglycerides. But the triglycerides, you swallow them, they get down into the stomach, there's no digestion in the stomach, and then finally they get into the duodenum. And so really digestion for lipids begins in the small intestine. So the triglycerides enter into the small intestine as large lipids. Okay, and then what's going to happen is that these large lipids, or these large lipid drops, are going to break down into tiny droplets. And how that happens is that as soon as the lipids are in the duodenum, this large organ here, which is called the liver, is going to secrete a substance called bile. And so bile will be, through a duct, will be deposited into that duodenum, and then the bile breaks those large lipid drops down into tiny droplets. And then after that, now we're at tiny droplets in the small intestine, and at that point, this other organ here, again, the pancreas, is going to secrete its enzymes into the duodenum, and the pancreas secretes what's called pancreatic lipase. Pancreatic lipase. And pancreatic lipase is going to break those tiny droplets down into fatty acids and monoglycerides. Okay, hope you can see that. Alright, so monoglycerides. And then the glycerol, which are monoglycerides, and the fatty acids can then get absorbed. Now again, those fatty acids and monoglycerides, just like the carbohydrates, are going to get absorbed in the second part of the small intestine, which is called the jejunum. But once it's in the jejunum, the other thing that's in those microvilli I showed you before were, we had these blood vessels, but the fats, when they get absorbed into those villi, they're actually going to be absorbed into the lymphatics. So they're going to get absorbed into a lymph vessel, because there's also a lymphatic vessel in here. Okay, so there's also a lymphatic vessel in here, and that's going to lead to a larger lymphatic vessel. And we know what happens to those lymphatic vessels. They head back up to the heart, and they dump into the subclavians. The fatty acids and monoglycerides get absorbed into the lymphatic system in the jejunum. Now, the last one that we have to talk about are proteins. And proteins, this is very time consuming. And it's also very complex. Okay? And what we know about proteins is that nothing is going to get absorbed in the mouth. Nothing's going to get absorbed in the esophagus. But protein digestion actually begins in the stomach. So in this organ right here. Okay? So we didn't have much digestion of anything else in the stomach. But in the stomach we have these cells that are called chief cells. Okay, and chief cells are going to secrete a substance that's called Pepsinogen. Okay, now Pepsinogen in a very acidic environment, and we know that the stomach has a pH of 1.5 to 2.0. If it is that low, then pepsinogen will get converted to pepsin, and then pepsin can break down proteins. Okay, and the pepsin will break those peptide bonds and degrade the proteins into polypeptides. Okay, now we said that the pH of the stomach is 1.5 to 2.0. And the reason why that pH is so low in the stomach is because of the other cells that are in there that are called the parietal cells. And those parietal cells are going to secrete HCl, hydrochloric acid. Hydrochloric acid drops that pH that was 4.5 in the mouth and the esophagus down to 1.5 to 2.0. Okay, so now in the stomach we have polypeptides, but that's still too big. We need to break those down even smaller. Those polypeptides are going to continue to get broken down in the small intestine, in the duodenum, because in the duodenum we have this, again, this pancreas, this great organ here. It's going to secrete more pancreatic enzymes, and this time the pancreatic enzyme is called pancreatic protease. Proteases break down proteins. Okay, and the pancreatic protease breaks these polypeptides down into even smaller dipeptides. Dipeptides. Okay, so now in the duodenum we have dipeptides. Alright, now at the base of the epithelial cells in the duodenum, there are some simple columnar cells that are going to secrete what's called a dipeptidase. And ACE, that tells us is an enzyme. Dipeptidase is going to break those dipeptides down into individual amino acids. Okay, and then these individual amino acids can be absorbed into the body. And they're going to get absorbed in the jejunum. Okay, so the second part of the small intestine is the jejunum. That's where everything so far has been absorbed. And they're going to get absorbed into the blood. And we know that all blood leaving the small intestine is going to go to the liver. That's the end of digestion and absorption of carbohydrates, lipids, and proteins.